respiration, we'll talk more about the mitochondria and the different parts, like what happens in each part. Your chloroplast, also two membranes, outer membrane, inner membrane. You have your thylakoids, which are these disc-shaped things, and inside the thylakoids is hollow, so that's thylakoid space. A stack of thylakoids is called a granum, and then surrounding all of that, you have fluid called stroma. And we learned about photosynthesis and how the light reactions happen in the grana, or the thylakoids, and the Calvin cycle happens in the stroma. We'll review that more when we review those chapters. Good? Again, chloroplasts have their own DNA. So more linkage to the endosymbiont theory there, or more evidence.
Own DNA.
Okay, peroxisomes, like I said, they're going to make hydrogen peroxide. The reason they're making it, though, is because it's a byproduct of breaking stuff down. It's not because, oh, I want to make peroxide, okay? And then the peroxide itself is toxic in high quantity, so that's going to get broken down into water and oxygen. Got it? So that's peroxisomes. Then we have the cytoskeleton. There are three parts of the cytoskeleton. Microtubules are the thickest, microfilaments are the thinnest, and then intermediate filaments are in between. Here's a picture with microfilaments. The microtubules are the bigger ones, microfilaments are the smaller ones.
What's the middle one?
The intermediate filaments.
Oh, wow.
Various jobs. For instance, they serve as kind of a subway system to move organelles around. Yeah. Microtubules. Microtubules make up the spindle fibers. So what are spindle fibers involved with? Mitosis. They help give structure to the cell, like keep the right shape for it. They also help this cell change shape. So if a cell needs to grow, then microfilaments are going to go in certain directions that cause the cell to elongate before the cell divides for cytokinesis. Okay? If you are an amoeba, your cytoplasm has to move around in order for you to grow, like the pseudopods, so the cytoskeleton kind of helps direct them. Okay, moving the stuff around is not something that is energy free, so you do have to use ATP to do this stuff, and you have special proteins called motor proteins that help out with that. So here's a picture. You've got your microtubule here. Let's say you need to move this vesicle from one side of the cell to the other. So it's like a little subway system or a monorail if you've ever been to Disney World. The motor proteins right here are going to use the ATP and like grab and pull kind of to get from one part to the next. So here's a picture taken with an electron microscope of that. Cool?
Isn't it instead of getting pulled towards it, like moves along?
That's the chromosomes along the spindle fiber. I don't think it works quite like that with this, though, because the E's stay in place.
That's another protein.
Okay? It's not a bad idea for you to know the name of the proteins involved. For microtubules, the protein is called tubulin. For microfilaments, it's actin. The other place that you have actin is in your muscles. Actin and myosin are the proteins in your muscles. Okay, so there's a good chart in your book. It's probably too small for you to really see here. But microtubules, again, tubulin and the types of things that it does. Cell division, that's the spindle fibers, helping the cell move around, keep the right shape, and moving the organelles about. We've already talked about all those. Good with microtubules? Microfilaments, also changing cell shapes. We said actin is found in muscles. Actin again. Some of the same stuff, so they work together a lot. Intermediate filaments. You probably don't need to know like specific jobs of these, just know that it's the intermediate size. Okay. Centrosomes and centrioles. A centrosome is a region. In a plant cell, the centrosome contains centrioles, but I said that backwards. In a plant cell, the centrosome does not have centrioles. In an animal cell, you do have centrioles. Their job is to organize mitosis. So they organize the spindle fibers for mitosis. Okay? The structure is that it's made of 1, 2, 3, 4, 5, 6, 7, 8, 9 triplets. Okay? I've seen picky questions like that, unfortunately. So centrioles, 9 triplets of microtubules. However, the cilia and flagella have a different arrangement. Does anyone remember what their arrangement of microtubules is?
5?
Instead of,
Something 4.
it's called 9 plus 2. So if we look at this, instead of triplets, we have doublets. 1, 2, 3, 4, 5, 6, 7, 8, 9 doublets, plus 2 in the middle. So it's a 9 plus 2 arrangement. Both your cilium and your flagellum have that same 9 plus 2
What are 9
arrangement.
triplets? Microtubules?
No, centrioles.
Centrioles.
Other differences between flagella and cilia. Cilia you typically have a bunch of, whereas flagella you'll only have 1 or 2. Flagella tend to be much longer, cilia are much shorter. Flagella beat side to side to propel you forward. And then in this one, these beat the opposite direction. So these are beating this way to go that way. These beat the direction parallel to the
Cilia move though? Like,
Yeah.
Doesn't they stay?
They're like beating.
Yeah, so they don't like actually move.
They don't come off.
Do they?
These go back and forth, whereas these undulate. Okay?
That's like
Where do you find cilia? Give me some examples.
Whatever that is.
You have it in your nasal passages. We talked about the epithelial tissue that has the cilia. It produces mucus. Not the cilia doesn't produce the mucus, but that nasal mucosa does. And the cilia are going to help move the mucus that's trapped debris so you can get rid of it.
so when your nose is like
You have flagella. In animals, it's going to be sperm cells is your best example. But in single-celled organisms, you have several different types of protists that have a flagella like Euglena does. And then cilia, you have things like Paramecium. I don't think you need to know this stuff. Myosin is a protein name that you should know that's part of muscles and we'll talk about muscles a little bit more tomorrow in class. We are going to do how muscles contract. So this is something I'll teach in class. Pseudopods and cytoplasmic streaming we talked about already. Do you have questions about that? That's the way things like amoeba are going to move around. Alright, cell walls we're familiar with. We already talked about which ones. You need to know the order that the cell wall parts are in. So you have your primary cell wall. Then you have your middle lamella, which is in between your primary and the secondary ones. You don't always have a secondary. And then the plasmodesmata are channels between. So looking at a picture, this is the area in between two cells. Okay? So if we look at this picture, the area in between two cells. Here's where we are. That's your middle lamella. And there's like a gluey kind of stuff there that keeps the cells together. Because you wouldn't want like part of the plant cell to come apart from the other plant. Like this cell just breaks apart. You need them to stay together. So that's your middle lamella. Here is secondary cell wall. Here is primary cell wall right there. That's the middle lamella. So then this would be primary cell wall of the next cell, and then secondary cell wall of the next cell. So primary is the outermost wall.
For animal cells, the outside, they don't like stick together, do they? They just,
there's a slide about that.
Where's the cell membrane?
It's the extracellular matrix that's in between all the cells. This picture's not show. It would be right here. The extracellular matrix. If we look at this picture, here is inside the cell. This is an animal cell. Here's inside the cell. Outside the cell, you have collagen, which we talked about in class, which is the most abundant protein found in the animal kingdom. We have different types of fibers that are going to be in this extracellular fluid, and they help anchor cells together. So it's not like a glue that sticks them together. It's more of things holding on to other things.
What's the integrin?
It's a protein that goes all the way through the plasma membrane instead of just being on the outside part.